Welcome to this comprehensive exploration of the accessory organs of digestion. While we often think of digestion as occurring within the gastrointestinal tract itself, three critical organs work from outside the digestive tube to make digestion possible. These are the liver, the gallbladder, and the pancreas. Each plays an essential role in breaking down the food we eat, and without them, proper digestion simply could not occur. Let's begin by understanding how these accessory organs connect to the digestive system. The liver, gallbladder, and pancreas all deliver their secretions into the duodenum, which is the first section of the small intestine. This connection point is crucial because it's where digestive enzymes and bile meet the partially digested food coming from the stomach. The liver produces bile, which is stored and concentrated in the gallbladder. The pancreas produces both digestive enzymes and bicarbonate. All of these secretions travel through ducts to reach the duodenum at precisely the right moment. Now let's focus on the liver and its production of bile. The liver is the largest internal organ in the human body, and one of its primary digestive functions is to produce bile. Bile is a complex fluid composed of water, bile salts, cholesterol, bilirubin, and electrolytes. The liver produces approximately 500 to 1,000 milliliters of bile each day. This bile flows through a network of ducts, first through the hepatic ducts within the liver, then into the common hepatic duct, and finally either into the gallbladder for storage or directly into the common bile duct toward the duodenum. The most important component of bile for digestion is bile salts. Let's examine how bile salts solve a fundamental problem in fat digestion. Fats are hydrophobic, meaning they don't mix with water. This creates a challenge because digestive enzymes work in the watery environment of the intestine. Bile salts are amphipathic molecules, meaning they have both water-loving and fat-loving regions. When bile salts encounter large fat globules, they break them apart into tiny droplets through a process called emulsification. This dramatically increases the surface area of the fat, allowing lipase enzymes to access and digest the fat molecules much more efficiently. Without bile salts, fat digestion would be severely impaired. Between meals, bile produced by the liver is stored in the gallbladder. The gallbladder is a small, pear-shaped organ located beneath the liver. Its primary function is to concentrate bile by removing water, making it up to 10 times more concentrated than when it left the liver. When you eat a meal, especially one containing fat, the hormone cholecystokinin, or CCK, is released. This hormone causes the gallbladder to contract, squeezing concentrated bile through the cystic duct and into the common bile duct, where it travels to the duodenum to aid in fat digestion. Some people undergo gallbladder removal surgery, a procedure called cholecystectomy. This is often necessary when gallstones form and cause pain or blockage. Gallstones are hardened deposits that can develop when bile contains too much cholesterol or bilirubin. After gallbladder removal, bile flows directly from the liver to the small intestine in a continuous trickle rather than being stored and released in concentrated bursts. Most people adapt well to this change, though some may need to modify their diet, particularly reducing fat intake to avoid digestive discomfort. Let's turn our attention to the pancreas. The pancreas is a long, flat gland located behind the stomach. It has both endocrine functions, producing hormones like insulin, and exocrine functions related to digestion. For our purposes, we're focusing on its exocrine role. The pancreas contains clusters of cells called acini that produce digestive enzymes. These enzymes are secreted into small ducts that merge into larger ducts, eventually forming the main pancreatic duct. This duct joins with the common bile duct at a structure called the hepatopancreatic ampulla before entering the duodenum. The pancreas produces a remarkable array of digestive enzymes, each specialized for breaking down different types of nutrients. For carbohydrate digestion, the pancreas secretes pancreatic amylase, which breaks down starches into smaller sugar molecules. For fat digestion, it produces pancreatic lipase, which works together with bile salts to break down triglycerides into fatty acids and monoglycerides. For protein digestion, the pancreas secretes several proteases, including trypsin, chymotrypsin, and carboxypeptidase. These are initially released in inactive forms to prevent the pancreas from digesting itself, and they're activated only when they reach the small intestine. The pancreas even produces enzymes for nucleic acid digestion, breaking down DNA and RNA from the food we eat. The secretion of these enzymes is regulated by hormones, particularly secretin and cholecystokinin. In addition to enzymes, the pancreas produces a bicarbonate-rich fluid that serves as an acid-neutralizing buffer. When acidic chyme leaves the stomach and enters the duodenum, it has a pH around 2, which is far too acidic for the intestinal enzymes to function properly. The pancreatic bicarbonate neutralizes this acid through a chemical reaction. Bicarbonate ions combine with hydrogen ions from the stomach acid to form water and carbon dioxide. This raises the pH to around 7 or 8, creating an optimal environment for digestive enzymes. The hormone secretin, released in response to acidic chyme in the duodenum, stimulates the pancreas to secrete this bicarbonate-rich fluid. The hepatopancreatic ampulla, also called the ampulla of Vater, is where the common bile duct and the main pancreatic duct converge before entering the duodenum. This strategic junction ensures that bile and pancreatic enzymes are delivered together at the same location. A muscular valve called the sphincter of Oddi controls the flow of these secretions into the duodenum, opening when digestion is needed and closing between meals to prevent backflow. The coordination of accessory organ secretion is a beautiful example of hormonal regulation. When food enters the stomach and then the small intestine, it triggers a cascade of hormonal signals. First, the presence of acidic chyme in the duodenum stimulates the release of secretin from intestinal cells. Secretin travels through the bloodstream to the pancreas, stimulating it to release bicarbonate to neutralize the acid. Second, the presence of fats and proteins in the duodenum stimulates the release of cholecystokinin. CCK has multiple effects. It causes the gallbladder to contract and release bile, stimulates the pancreas to release digestive enzymes, and even slows stomach emptying to allow more time for digestion. This hormonal coordination ensures that digestive secretions are released at precisely the right time and in the right amounts. Understanding these organs also helps us appreciate various clinical conditions. Gallstones, as we mentioned, can block bile ducts and cause severe pain. Pancreatitis is inflammation of the pancreas, which can be acute or chronic and is often caused by gallstones or excessive alcohol consumption. When the pancreas becomes inflamed, its digestive enzymes can begin to digest the pancreas itself, causing severe damage. Hepatitis refers to inflammation of the liver, often caused by viral infections, and cirrhosis is scarring of the liver tissue, commonly resulting from chronic alcohol abuse or hepatitis. Cystic fibrosis, a genetic disorder, affects the pancreas by causing thick mucus to block pancreatic ducts, preventing enzyme secretion. Pancreatic cancer, though relatively rare, is particularly deadly because it often goes undetected until advanced stages. Let's walk through what happens during a typical meal to see how these organs work together. First, when you eat, the sight, smell, and taste of food trigger the cephalic phase, preparing your digestive system. Second, as food enters the stomach, gastric acid begins breaking down proteins. Third, when acidic chyme enters the duodenum, secretin is released, triggering bicarbonate secretion from the pancreas to neutralize the acid. Fourth, fats and proteins in the duodenum trigger CCK release, which causes the gallbladder to contract and release bile, while also stimulating pancreatic enzyme secretion. Fifth, bile emulsifies fats, while pancreatic enzymes break down carbohydrates, proteins, fats, and nucleic acids. This synchronized process ensures efficient digestion and nutrient absorption. Beyond its digestive role, the liver performs numerous other vital functions. It processes nutrients absorbed from the intestine, converting them into forms the body can use or store. It detoxifies harmful substances, including alcohol, drugs, and metabolic waste products. The liver stores glycogen, vitamins, and minerals, releasing them when the body needs them. It also produces plasma proteins essential for blood clotting and immune function. The liver truly is a metabolic powerhouse, performing over 500 different functions. To summarize, the accessory organs of digestion are essential partners in the digestive process. The liver produces bile, which is stored and concentrated in the gallbladder and released to emulsify fats. The pancreas produces digestive enzymes for all major nutrient types and bicarbonate to neutralize stomach acid. These secretions meet at the hepatopancreatic ampulla and enter the duodenum together. Hormones like secretin and cholecystokinin coordinate the timing and amount of these secretions. Understanding these organs and their functions not only helps us appreciate the complexity of digestion, but also helps us understand various digestive disorders and their treatments. Thank you for joining me in this exploration of the accessory organs of digestion.